In the name of God, the most gracious and the most merciful. I am محسن, Assistant Lecturer at the Department of Microbiology, Faculty of Medicine, University of Babylon. Our lecture today is the resurgence of pediatric rotavirus infection, and this lecture is given for the third-year medical students. Our lecture today is considered to be as both fascinating and essential, as it focuses on the comprehending the fundamental pillars regarding rotavirus, including its structure, mode of transmission, pathogenesis, and the maternal antibody protection as a form of immune response, the laboratory diagnosis, and to delineate the most effective method for diagnosis and identifying this tiny agent, and eventually a treatment including vaccine, and the prevention measures that we should dealing with them in order to mitigate the burden of disease. So we have a lot to unpack today. So let's jump right in and let's get started. Rotavirus is considered to be as the main etiological agent that is responsible for causing an inflammation in the intestine and the stomach, and is responsible for causing millions of cases annually around the world, especially in infants and young children less than 5 years of age. And this concentration of the infection in this age group can be attributed to many factors. I will allude to them during the flow of my speech. This virus is belong to Reoviridae family, and it has been estimated that the virus particle is about 76.5 nanometer in diameter. And there are at least 10 species of rotavirus that are distributed around the world. The rotavirus A, it is considered to be as the most common for a human. This virus is non-enveloped virus, or it is also called naked virus. The non-enveloped virus renders the virus very stable in the environment and render the virus at the same time to be resistant for chemical and physical agent. And it has to be noted that the segmented viruses allow for reassortment. As we know, this virus is segmented, have a segmented genome. And there is an important phenomenon which is also called reassortment. So in this animation, as we can see, this is a virus infected cell. When two different strains of the same virus co-infect the same cell, during viral replication, the RNA segments are mispackaged mistakenly during the replication and lead to emergence of a new strain that is totally different from the original ones. This is called to be as reassortment. What is totally happened is a form of genetic mixing. And also this virus coding for structural and non-structural proteins. And the main difference between structural and non-structural proteins is that the non-structural proteins are expressed from the rotavirus infected enterocytes, while the structural protein are normally a part of the virus composition. This is the rotavirus structure. Rotavirus is also called to be as icosahedral. The symmetry of virus is icosahedral, triple layered capsid. And there are 3 layers. We will identify each one of them intensively. Viral protein 1, viral protein 3. The viral protein 1 and viral protein 3 are located at the vertices, as we can see, are located at the vertices of the inner core, while the viral protein 2 form the inner shell of the core, while the viral protein 4, which is considered to be as the major glycoprotein by which the virus interacts on our cells, it's considered to be as a spike that is located on the outer surface of the virus, and the viral protein 6, which form the intermediate capsid, add to that, and eventually the viral protein 7, which is a protein-sensitive protein that is located and form the outermost layer of the virus. The mode of transmission. Rotavirus has the ability to transmit from person to person and has the ability to transmit from or via fecal oral route. So the ingestion of virus can be occurred from contaminated hands, food, and even water. So it is common in areas with poor sanitation and poor hygiene. And also this virus can be transmitted from one person to person by contact with feces, as I have mentioned, and at the same time, especially in the daycare centers and the hospital. And as we know, the infection that can be acquired in the hospital is called to be as nosocomial infection. And this virus has the ability to spread by caregivers or children through unwashed hand. And also the fomite transmission is usually occur in this case of rotavirus, because rotavirus, as we have mentioned, is non-envelope virus and can survive on a surface for multiple days, because the lack of envelope render the virus resistant to physical and chemical agents. Let me stress the significance behind this critical key point is that the pathogenesis of virus, basically the entry of virus occur through mouth, and then the virus attach to its own receptor in the small intestine, principally in the enterocyte. And it has been illustrated that the receptor with which the virus interacts on our cell is thought to be as the sialoglycan along with co-receptor integrin alpha-2 beta-1 by using a viral protein 4 or the glycoprotein. Then after replication of virus in the enterocyte, start to express its non-structural protein, and the main protein or the main non-structural protein who is responsible for causing all of these clinical manifestations is regarded to be as the non-structural protein 4, because non-structural protein 4 acts as a viral enterotoxin. Non-structural, what entirely and what precisely non-structural protein 4 does in the enterocyte is that the result in the increase in the intracellular calcium, and then result in the disruption of calcium homeostasis and disruption in the epithelial cell homeostasis, and then the increment of calcium can activate the chloride secretion, chloride ion secretion, and at the same time inhibit the sodium secretion, which lead to a case which is also called hyponatremia. And at the same time, the increment of calcium can lead to inhibition in the glucose transporters. And it has to be noted that the non-structural protein 4 play an important role, or play an indispensable role in the disruption of proteins who are responsible for the tight junction between enterocytes. These proteins are called to be occludins or claudins. So the non-structural protein disrupt these proteins and result in the dissociation of enterocytes from each other. And this is the main point that the non-structural protein 4 done. And then result water diarrhea, which is non-bloody. This is the first manifestation regarding diarrhea. Regarding the activation of enteric nervous system by also non-structural protein 4, which may lead to vomiting and nausea. The non-structural protein 4 play an important role to stimulate enterochromatin cells. These enterochromatin cells that are present, as we can see in this figure, are present in the intestinal mucosa. So stimulate these cells to secrete serotonin, which is also considered to be as neurotransmitter, and or also it can be called as 5-hydroxytryptamine. And the release of serotonin can lead to certain ramifications. On the top of the list is that the release of massive or the excessive release of serotonin can stimulate the vagal afferent nerve, and as I have mentioned, the serotonin play an important role in the stimulate the smooth muscle contraction indirectly by neurons. These neurons are called to be as intrinsic primary afferent neuron and lead to, at the same time, stimulate the intestinal secretion of electrolyte and the fluid, and then play an important role in the flushing out of harmful agents that may present in the small intestine. And finally and eventually, the excessive release of serotonin due to the non-structural protein 4 aid in the vomiting, because due to the inflammation or due to the aggregation of these viruses in the small intestine. This diagram show the maternal antibody protection. As we can see, the ingestion of secretory immunoglobulin from breast confer protection. Confer protection because the ingestion of secretory immunoglobulin A add to that immunoglobulin G and immunoglobulin M. The secretory immunoglobulin A bind to the receptor with which the virus in the normal case binds, and then block the interaction of virus on its own cell, and then confer protection to the child. And it has to be noted, according to the recent research, it has to be mentioned that the suckling infant are less vulnerable to be infected by rotavirus infection. The lab diagnosis Detection of rotavirus can be done by using the stool specimen or urine specimen by using certain methods. The most common method and the most reliable and sensitive method also we can consider it as the immunochromatographic rapid assay. Immunochromatographic rapid assay by taking a small drops or by taking a few drops from the urine and put these drops on the slide or this cassette, and within a few seconds, not exceed 30 seconds, the result will be positive, and the red line indicate positive result. Regarding the another method, the real-time PCR, we can use this real-time PCR, but this, although this method is sensitive, robust, and reliable, that this is considered to be as time-consuming and give accurate and specific result. As we can see in this picture, this is a test for a child suffering from acute gastroenteritis as a result of rotavirus infection. The red line indicates positive result for both stool and urine. As you can see, the C indicate for control, A indicate for adenovirus, and R indicate for rotavirus, and S is the place where the specimen is placed. And eventually, the treatment and prevention. No specific antiviral to this date is present against rotavirus. And the fluid replacement for severe cases such as dehydration should be done, because dehydration it is considered to be as a fatal condition for the infant or for the children. Hygienic measures such as proper hand washing should be take in our consideration, because this mitigate or reduce, as I have mentioned in the first slide, mitigate the burden of disease. And regarding vaccination, there are two different types of vaccination, Rotatec and Rotarex. Both of these vaccines are given for the child by putting certain drops or a few drops in the child's mouth and then confer protection against such tiny agent. This is it for today. Thank you for your listening, and I'm grateful for the Babylon Importance Multimedia Center for granting me this opportunity to present this lecture. Thank you.